Hello, good afternoon, everyone. Um, my first time here, and it's my first time being back to the United States after 4 years. So it's nice to be back. I miss the cold weather. All right. Now, this afternoon, I'll be talking about a model of plasmid segregation. So essentially, what we'll talk about is a plasmid, which is an extra chromosomal piece of DNA. You could imagine it to be a piece of DNA, circular, and then which is floating or diffusing in the cytosol of a cell. Now, um, we would talk about the case of col E1, which is a type of plasmid, which is high copy plasmid. Usually, the copy number is around 30 to 50, and that it is replicated in a process by which you have an RNA2, which is a pre-primer. And in the process of replication, this pre-primer gets attacked by RNA1. And if it's get attacked, they form a kissing complex. If the kissing complex is stable and it goes through the entire process, then replication doesn't happen. Now, if it doesn't get attacked in this inhibition window, which is base 110 to 10360, then replication happens. That's the case of col E1. Now, the col E1 plasmid is high copy plasmid, and in the case of high copy plasmids, they are thought to be freely diffusing in the cytosol. And in these cases, there's no active partitioning system for plasmids, in which case you would imagine the segregation to happen binomially. And therefore, plasmids with high copy number would usually have higher or lower probability. If it's high copy, then you've got low probability of having the plasmid not being segregated to one of the copies after, or to be lost, for instance. So you could imagine a cell, an E. coli, dividing into two. Now, in some cases, segregation happens and that all plasmids will be segregated in just one of the poles. The more plasmid copy you have, the lower the probability of this plasmid being segregated to only one of the poles. We call that loss probability. And for high copy plasmids with copy number of around 40, this loss probability is around 10 to the negative 12. But the motivation is that if you would be looking at experiments, the loss probability is several orders of magnitude higher than what is predicted by binomial segregation. So this is around 10 to the negative 6. So our question is, what drives that difference? And one possibility is because of the aggregation of plasmids or clustering of plasmids. It has been seen in experiments that plasmids do segregate and that they localize preferentially at the poles. And that will be the motivation for our model. So we formed or we constructed a mathematical model, a stochastic model, which accounts for plasmid segregation with clustering of plasmids. That's essentially the idea. And we considered the following elementary processes. Number one is replication. Now, replication happens for any of the plasmids inside the cell. And this replication, as was described earlier, has a probability of survival. So by survival probability, that RNA2 doesn't get attacked by RNA1. And that probability is given by the following. And the numbers there, 602, is due to Avogadro's number. So what happened is that we translated a loss probability, which is continuous in time or continuous in state space. We translated it into a discrete state space model. So what we got is this probability. A2 there is the transcription rate of RNA2. Alpha 1 there is the transcription rate of RNA1. And N and S0 here are parameters that we fitted. N is the sensitivity of plasmid replication. And then S0 is a threshold for RNA1 concentration. Right? So we've got this probability. And the first one is plasmid replication. The second is plasmid diffusion. So what happens here is that a plasmid could transfer to one of the poles. If it's in pole 1 or the old pole, it could transfer to the second pole or the new pole. And in our case, we look at plasmids in two ways. Either a plasmid is free or a plasmid is clustered. Only free plasmids can diffuse from one pole to another. Clustered plasmids do not get to transfer. They are localized at the pole where they are in. That's the first one, plasmid diffusion. Second one, rather, plasmid diffusion. We've got cluster dissociation. In our case, we assume that dissociation happens by shedding only one plasmid at a time. So from a cluster, a plasmid is shed, and now you have dissociation. And finally, the third one is cluster formation. So cluster formation can happen in two ways. You've got two clusters combining, or two free plasmids combining. So you would imagine that clustering and diffusion now would control the mechanisms of plasmid loss. The plasmid loss now could be affected by these two, or essentially by all four elementary processes. And we model plasmid segregation in this way. So imagine you've got a newly divided cell. We assume that all plasmids are inherited in one of the poles. We call it the old pole, either clustered or free. That's what you're seeing on top. And in the process, in the cell cycle, plasmid replication happens, diffusion happens for the free or mobile plasmids, and clustering may also happen. And after the cell cycle, you've got plasmids in the old pole and plasmids in the new pole. Cell division happens, and we track one of the daughter cells. And we try to estimate the loss probability by looking at a lineage and keeping note of the number of generations before we would see plasmid loss. And in our case, the plasmid loss probability is seen to be the parameter of a geometric distribution. All right, so that is the parameter P, which is the plasmid loss. And in our model, we're able to track the following at the singular cell level. So we could track the total number of plasmids, which is the black curve. And then we also could track the number of plasmids at the old pole and the number of plasmids at the new pole. Now you would notice that because we assumed that all plasmids get inherited in the old pole, typically you will see a larger number of plasmids there. And that because the number of plasmids in the old pole typically starts 0, there's nothing in there, so you will see a lesser number of plasmids. All right? Now we tried to fit the model using data from published data. And in this data, you would see that as division time increases, the copy number decreases. And in fact, these are the numbers. The division time, 100 up to 24, this is in minutes. And then the average plasmid copy number increases from 39 to 55. Division time could also view it as a decrease, or as the increase rather in growth rate in this direction. And what we did was to try to fit our replication probability using this data, trying to match the number of the average plasmid copy number with predictions of our replication model. And we made use of ABC, or approximate Bayesian computation. Our model essentially is this replication model, and then we try to match the last, or the copy number at the end of cell division, or of the cell cycle. And they were matched to these average copy numbers. You would see at figure A, some samples from ABC. And in figure B, you would see here the distribution of copy numbers of plasmids before cell division for 3 different regions of our posterior distributions, or the posterior samples. So you would see here N from 2.5 to 3. And previous estimates of this sensitivity index N is around 2.5 or 2.4. So we are at the ballpark. Okay. Now, by the way, I wasn't able to say this, but other parameters in the replication model also change with division time. Included here are the transcription rates for RNA2, RNA1, and the volume of the cell. They also change in time with respect to division time. We also tried to fit the clustering parameters by ABC by matching our model predictions with distribution found in literature. And among those are the following. So we've got data about the ratio of inherited plasmids. Green here is simulated versus experiment. And on panel D, you would see here the distribution of the number of large clusters within a cell. And we were able to match this quite closely. All right. Now, the main question really is, how does clustering affect the loss probability? All right. And first, to make this comparison, we have a base model looking at a model where clustering is absent. And you will see here that in the absence of clustering, the distribution of plasmid copy numbers become quite consistent from birth to the end of cell division, while the addition of a clustering mechanism increases the variance in the distribution of the number of plasmids inherited at birth. And as the cell cycle continues, this variance is actually corrected, and this variance this variance becomes smaller in time. So here you see that in the second panel, or the second column of the figure. So clustered plasmids segregate as groups, which increases this variability that you would see in the green panel. And without clustering, copy number variance becomes nearly constant. You would see there at the purple. And clustering inflates variance, while copy number controls or corrects it over the cell cycle. That's number one. Next, so we try to look at what happens when we increase the clustering rate. And if we do so, the first thing you would notice is that the number of free plasmids decrease. And from left to right, that will be increasing the cluster binding rate. We call it beta. And in the old pole and the new pole, you would see a decrease in the mean and the overall distribution. So that's number one. Because the number of free plasmids decrease, and the free plasmids are only the plasmids which could migrate from one pole to another, you would expect that asymmetry of plasmid distribution increases. By asymmetry of plasmid distribution, we mean the number of plasmids in the old, or the difference rather, in the number of plasmids in the old and the new pole. Now remember, all plasmids are inherited in the new pole, in the old pole rather, and you have nothing to start with in the old pole. And the number of plasmids in the new pole only increases once one free plasmid gets transferred there, and replication happens. All right. And furthermore, so with the decrease in clustering rate, there's increase in asymmetry. There is also increase in loss probability. And we would argue that this asymmetry increase is what leads to the increased loss probability that we see, or that could explain rather the difference in experiments and what is predicted by binomial distribution. So it's still clustering binding rate is increased here from left to right, and you will see an increase of loss probability from 10 to the negative 7 to around 10 to the negative 5. And this effect is countered by this is what one would expect, that if we increase the shedding rate, we would also see a decrease in the loss probability. Right. Now, we have seen earlier that the copy number depends on the division time. The higher the division time, the lower the copy number is. So logic would tell you that, again, we were saying earlier that as the copy number increases, loss should decrease, right? Now, what we found is that this is not necessarily the case. So now first, initially, we tried to match our model predictions with one which you would expect in binomial segregation. This is division times 24 minutes, 30, and 40. And what you would see here is a copy number before division distribution. And we looked at faster growth, which means lower division time. And recall that with faster growth or lower division time, copy number increases. So that's from left to right, you would expect that copy number would decrease rather. And then also the time available for diffusion decreases. Now these two things are countering effects because number one, an increase in higher copy number would push the probability lower, but the time available for division would push the loss probability to be higher, right? We tried to investigate this, and this is the model prediction. To the left, you would see here the prediction of our model with put against the prediction of a binomial distribution model. So essentially, when we say binomially segregated, the plasmids would be put into one of the poles with 50-50 probability, which means that the loss probability is around 1 over 2 to the n minus 1, n being the number of plasmids, right? So from left to right, the binomial distribution the binomial model would predict that you would have an increasing loss probability. But our model predicts a different trend. Our model says it should decrease. Now, which one is correct, you would say? Now, all right, so let's just say that. So now we try to investigate. First, so let's just not say which one is correct. Let's not ask that yet. So we try to investigate why this happens. Well, the trend for the binomial distribution is easy to explain, right? But the opposing trend given by our model is quite difficult. Now, so what did we do? So we proposed a mechanism for this. To be able to do so, we looked at a simplified model, an ODE model, for which we view plasmids as either free or clustered. We would call N1 the total number of plasmids in the old pole, where everything is inherited initially. And then N2 being the number of plasmids in the new pole. The number of plasmids is the total of free and clustered plasmids. Free is F here, B is clustered. And that the number of free plasmids decrease when they become clustered, and they increase if shedding happened. So there would be transfer rates nu and in kappa here, and that only the free plasmids could transfer between the poles with rate rho. All right, now. Okay, now, you would see here in the panel called D that the change of the number of the difference between the number of free plasmids decreases much faster than the delta n, which is just n1 minus n2, which is the asymmetry essentially. And because of that, we were able to make a quasi-steady state approximation for delta n, and it looks like this. And you will see here that d delta n, or delta n, decreases exponentially, which is promoted by rho and kappa, which is decreased by the transfer rate, or by the clustering rate rather, nu. All right. So now what does this tell us? If you would be looking at the red curve right there, you would see that longer division times produce plasmid asymmetry by allowing more equilibration before division. And elevated loss at fast growth is driven by clustering-mediated asymmetry, but not lower copy number. And that essentially is our proposed mechanism for it, asymmetry, brought by clustering. And now what we ask, is this true? Is this something that we could observe from experiments? So what we did was to design an experiment looking at 3 possible division times as 30, 35.7, 52.5 from top to bottom. And then the copy numbers are given in panel C. So for the top one, the fastest growing, it's around 50. The second fastest growing is around 40 copy numbers. And then the last one has copy number around 30. Indeed, we were able to observe the trend that is predicted by our model. that if you would be increasing division time, you would see a decrease in loss probability as given by the experiments, which was not observed before. And with that, we say that within the parameters that we explored, that our model is correct and it is able to predict some trends. All right. And as conclusion, in our work, we constructed a stochastic model, discrete state space model, which is because you have there a non-autonomous part. It is a non-homogeneous CPMC, and the prediction is that clustering increases plasmid loss probability by increasing asymmetry of plasmid distribution. And the model predicts that plasmid loss is decreased with longer division time. And this model predictions were verified experimentally, showing that cells which divide faster are more likely to lose the plasmid. And thank you for listening, and special thanks to Dr. Krasiosic, Dr. Oleg Igoshin, Harris Yanitsis, Amanda Alexander, and Dr. Matthew Betten. Thank you.
Jayson? I have a question, which is, what's the benefit then of clustering? Like, it sounds like clustering, at least from this vantage point, is just bad. So what's going on?
That is actually something that we were trying to explore. Initially, we were trying to look at what is the evolutionary benefit of having clustering. And right now, we actually don't have an answer for it. But it is something that can be explored in the future. But really, there must be something about it because it happened in nature. So there must be a benefit for having it. But right now, we don't have any explanation for it. Yeah.
I suppose once clustering is happening in the new pole, that's beneficial in the sense that it's maintaining some symmetry, or it's maintaining mass in this like new pole that started with nothing.
Well, that's true, but clustering, if you would remember, would increase loss probability.
Right, exactly.
Which means we lose some characteristics. For instance, because plasmids, for instance, could give a cell resistance to antibiotic. It gets lost in that case. So really, it's a mystery why we have it.
We did just speculatively. We talked about this. What could happen is that this could be regulated clustering, and you could turn it on, and then just a subpopulation of the total population would inherit the plasmids,
yeah,
and the potential for antibacterial resistance would just be carried by a small subpopulation until it's needed, right? And they carry the burden because plasmids are metabolically expensive.
Yeah.
So the subpopulation carries the burden, and then once you need it, they can actually then also share the plasmids with others, so you have some segregation.